Hello, hello.
Loud and clear.
That red light is shining. Okay. Well, I will, and they should be able to hear you because if you speak into it now. Okay.
Few minutes.
And so you're ready to speak, just hit that button up there.
All right, this is a nice room full of people. Can everybody hear me? We were just making sure that our Zoom programming was working. So it is now. We do have some people joining us on Zoom, so we want to welcome them as well. My name's Mayo Taylor. I'm president of the Blue Ridge Naturalist Network. And this fall, we are celebrating our 10 years of existence. And this makes me feel like we're pretty robust to see all of you out here. So thank you for being here tonight. We also have about a dozen people on Zoom. And we do a few things differently for them when we kind of hold the questions till we get to the end. And then anybody who wants to speak has to be up here or have a microphone brought to you at your seat so that people on Zoom can hear what we're doing. And then people on Zoom, if you want to send in questions by chat, we'll be taking those and putting them out as well. Well, I always like to start by thanking Ollie and the Reuter Center for hosting us here. I want to particularly thank our student tech, Tyler. We would not be able to do this sophisticated hybrid presentation if we didn't have the support of Ollie and Tyler. And they do support a number of other organizations. We've got someone here from the Astronomy Club tonight, and they meet here. And I'm supposed to tell you that on the first Wednesday of November, there will be what sounds like a pretty fascinating program on the French Broad River. And there's an effort underway to have its natural features recognized by getting a wild and scenic designation. So other announcements. We are doing one more indoor program after this. It will be on the second Tuesday again in November. That's November 14th. And the topic will be coyotes in Western North Carolina. And our speaker is going to be Justin McVeigh, who is the district biologist for this area. And he's with the North Carolina Wildlife Commission. And he is considered a megafauna expert. And he did his graduate work on coyotes and red wolves. So it should be a really interesting program. I hope a lot of you will turn out. And I'm going to call up Paul Fredrickson. Paul's on our board. He's an incoming outdoor program leader, and he's going to tell you about a hike.
Thanks, Mayo. Yeah, we have a wonderful event next Wednesday, the 18th, and we're going to combine two things. Number one, we're going to be looking at the beautiful trees that are going to be turning color, and also we're going to be visiting two waterfalls. I'm not sure we're going to see any wildflowers. We might, maybe the residual of some of them, but what we're going to be looking at are waterfalls along 475. If you're familiar with Pisgah, it's by the fish hatchery. That's 475. That's the road that goes by the fish hatchery. and we'll be going there. And the first one will be Cove Creek Falls. It's about a mile, maybe 1.2 miles, pretty gradual hike. And then we'll be hiking down to the falls, the base of the falls, and then coming back. And then we'll be driving a little bit further on 475, and we'll be going to Tom Springs Falls. That's a pretty gradual walk, about half a mile. And the falls are right by the side of the road. It's pretty impressive. Not a whole lot of water probably at this point, but yeah, who knows? Who knows? Maybe this weekend. But it's an amazing drop. It's about 100-foot drop of cliff there. Anyhow, if you have any interest in either one of those, let me know. We're going to be going out there, probably arriving at the trailhead, the first one at 10:30 in the morning on Wednesday, and then probably having lunch and then be back sometime mid-afternoon. If you have any interest, please just reach out to me after the meeting and I'll put you down. Okay. So very good. Thank you, Mayo. And I think I have you signed up, don't we? Excellent. Thank you.
Since I know a lot of you who are here tonight are not members, if you and probably do not get our monthly newsletter, if you would like to get our emails about events that we do, including these indoor programs, there's a sign-up sheet at the end of a table over here. Just give me your name and your email and we'll get you in the system to do that. Well, with that, I'm going to bring up Diane Mooney, who is our communications guru. And she's the one who found our speaker and is going to do the introduction.
Thank you. Tonight, our speaker is Jenny Gruhn, PhD, owner of Asheville Hiking Tours. Jenny got her biology degree at UNC Chapel Hill. For 3 years, she researched tropical plants of Ecuador, Colombia, and Peru. And for her PhD, she investigated Rocky Mountain and Sierra Nevada pine forests. She is a certified interpretive guide and has led nature tours for citizen science programs and clubs of nature enthusiasts. We welcome you, Jenny. Thank you.
Hello. Are we good? Okay. Bear with me with the mic. So lovely seeing so many faces here today. Thanks for coming out. It was just my mom sitting here for the first 20 minutes, and I thought it was just gonna be my mom tonight. I was okay with that, but so yeah, my name is Jenny Gruhn, and I do own Asheville Hiking Tours, but by training, I am somewhere between a evolutionary biologist and conservation biologist and forest ecologist. It's kind of hard to say because research is so interdisciplinary, of course. So I'm excited to talk to you about research that I did. It is a little bit dated, but it's timeless research because I got my PhD maybe 7 years ago, almost now. Originally, I am from North Carolina. It all started in Waxhaw, Weddington area. Thanks, Mom. And so that's in the Piedmont, of course, at the base of the mountains. But oh, like that? Is that better? Like that? Check, check, check. Is that better? Okay. Thank you. Yeah, that is kind of annoying. I spent a lot of time in the Appalachians nonetheless growing up. So the rite of passage, of course, if you're growing up in North Carolina, you go to Sliding Rock every summer. You go to camp in Hendersonville, North Carolina. These are things I did. Your parents buy a mountain house. That's something they did when I was in high school. So I went to the mountains of western part of Virginia quite a bit. The east side of Grayson Highlands area. It was just gorgeous. I spent so much time there. It's dear to my heart. And of course I went backpacking. I was a trip leader in undergraduate, as an undergraduate for backpacking. And probably a more formative time during my undergrad days was at Highlands Biological Station. So I took classes by Peter White and Thomas Wentworth in conservation biology and forest communities of the Appalachians. Did some research in the Smokies with Peter White. And by the way, I'm going into my history just because I know I'm horrible at networking and a lot of you don't know who I am. So I'm just gonna dive right in and explain why I'm here today, where I came from. And eventually, I went and studied abroad in Ecuador during my undergrad days, and I fell in love with the Andes. You know, coming from the Appalachians and then going to the Andes, I just loved mountain systems in general. And so, as my eventual boss told me, I weaseled my way into Missouri Botanical Garden. And I volunteered for so many different people, I eventually got a job. But it wasn't just any job, it was my dream job working for the Ecuador program at Missouri Botanical Garden. And this meant that I got to be part of teams on pack trips, identifying, collecting, describing new species. And we would average one new species every 3 days out of Ecuador. I got to be part of the process of naming them. Maybe I got one named after me, but that's not important. So yeah, we did expeditions, and these are National Geographic funded ones in Colombia, Peru, and Ecuador. Absolute dream job material. Um, and then Peter Raven was the director of Missouri Botanical Garden. If you ever took a botany class, he probably wrote your textbook. And he gave me a lot of good guidance in those days, particularly when he became an advisor of mine when I went to Washington University in St. Louis. So I decided to stay in St. Louis. For those of you who don't know, St. Louis is a hub for botany. People flock there to study not only tropical environments, but temperate environments. And it's because the Botanical Garden, thanks to Peter Raven, it employs 500 botanists all over the world. Madagascar, China, South America. And these are people doing just top-notch conservation research. And those were very special years at Missouri Botanical Garden. And now here I am back in North Carolina. And so after going to Washington University in St. Louis, I realized there weren't a lot of jobs available in Western North Carolina, so I decided to make one. And I always loved teaching field biology the best as an undergrad, and I thought, why aren't we doing this with adults for fun? Like, people should be paying for this, right? So I started what's essentially an eco tour company, and a few of you in the audience were on this tour. This is one of our community hikes, and this was a birding trip we took just this past summer up on the Parkway. And I think there's 3 of you in the audience on this tour. That's fun. And so yeah, we do community hikes like birding and wildflower walks. We did a geology tour a few weeks ago. I did a communities hike 2 weeks ago. And of course, our bread and butter tours are for the general public. We run at least 2, usually 3 vehicles out to the Parkway and into the National Forest every day. I have a staff of 10 people altogether. Some people are here, thanks. And yeah, we have a lot of fun with it. I only employ people who are true naturalists or biologists, even if they're self-taught. You know, they have to be very passionate about local flora and fauna. And so we have very engaging tours, and we're practicing ecotourism at its best by being a donor to Southern Appalachians Highlands Conservancy and giving back to the Parkway and the National Forest through our permitting process. So yes, that's a little bit about Asheville Hiking Tours. So now, what are we going to go into? Well, I'm really excited to talk about something that's so passionate. So I am so passionate about distributions in general, about spatial patterns in the landscape, whether that's species or like traits of species. And a lot of this comes from having done research in South America in the Appalachians. And then as I'll go into, research for my PhD was out west in North America, so in the Rockies and Sierra Nevada and the Cascades. And I always worked in mountain environments. I love the mountains. We all understand that, right? And so I'm deeply interested in patterns that you see in mountains, like elevation gradients. And some of this comes from having been in these disparate environments, and yet you see these patterns emerge. Like, for example, when you're on a rocky outcrop in high alpine Colorado, and you're on a rocky outcrop in Colombia and here, you always are going to see a little stone crop plant, right? You always see plants with thick, leathery leaves and with lots of hairs to protect them from the radiation. And so when you see these patterns, like seeing ericaceous plants in acidic soils, right? Or oaks and basic soils or what have you. So basically, I'm deeply interested in these spatial patterns. Some people said that's because in grad school, they used to tease me and say I think like an artist and not a scientist. So yeah, I like distributions. So now plant distributions, of course, are going to be dictated, where they're occurring, plants are going to be dictated by so many environmental variables. You have climate being a major one. There's also, competition from other species that'll prevent a plant from being somewhere. There's mutualisms that encourage plants to be somewhere. There's the soils, what have you. But more so than any other predictor, climate, so precipitation and temperature combined, is going to determine where we see species. And for this talk, I'm focusing on plant species, of course. So this is a world map of climate classification. And climate, as I said, is based on temperature and precipitation. It's largely the coldest weather or the very hottest weather or the wettest weather that's going to dictate species boundaries. So where you expect things to have their distributions cut off. Now, interestingly, the climate has changed just a little bit in Earth's history. So in the first part of my talk, I'm going to be going into deep history. So evolutionary history that dates back to 60 million years ago, 80 million years ago, up until the Pleistocene, which started about 2.5 million years ago. That I'm going to call more recent climate history, even though it's still 2.5 million years ago. So the first part of my talk, I've divided up into chapters, is going to be ancient climate. And then the second is going to be more recent climate, both impacting species distributions of plants. So, who remembers Pangea? Did you all learn about Pangea back in the day? Yay! Some people are shaking their heads no. So yeah, plate tectonics really hasn't been around that long. The theory of plate tectonics has been around only since the 1970s. But yes, the Appalachians were actually formed when Pangea was formed. The central Pangean mountains gave rise to the Appalachians, the Scottish Highlands, mountains in Morocco, Africa. It's pretty wild. Again, these big worldly patterns, right? They're mind-blowing. And then Pangea started to break up to 225 million years ago. And it's still breaking up today. So this is present day right here. And a time I want to focus on for the purposes of today is about 50 to 60 million years ago. The Earth was extremely hot at that time. So we have fossils of crocodiles and palm trees found in Greenland at that time. We're talking tropical northern latitudes, no ice sheets like we have today. And interestingly, the continents were a lot closer at that time too. So you had these land bridges connecting the continents. Now this is an image looking down on Earth from the top. And so here we have Asia, rather connected to Europe, connected to North America, and this is the Beringia, of course, between North America and Asia. And so again, Earth is hot, and this it was a lot hotter even like 60 million years ago. And what I'm going to try to explain to all of you in as few boring graphs as possible is a kind of complicated concept, but basically the flora of Asia, Europe, and China used to have common ancestors at the northernmost latitudes. Millions of years ago, before things evolved into what we see today, the common ancestor, the Adam and Eve, lived in the northernmost latitudes. That part makes sense, right? um, and that is why if you walk through the forests of China today, it feels like you're in North Carolina. Because you are walking through forests that are the cousins of the species that we have here. They all descended from those northernmost latitudes when Earth cooled and species were pushed down southward. As it was cooling, it was getting so cold that the species are like, I'm out of here, I'm going south. And they occupied Southeast Asia, particularly China and the mountains there. Mostly the southeastern parts of North America, because we have more species here, but all through North America, even into South America, they went. And they occupied Europe. So does that part make sense so far? Okay. So they're tracking southward. Now, I focus on Asia here because in Europe, most of the species that tracked southward in Europe, they went extinct. So just 21,500 years ago, we had our last glacial maximum. And again, that's recent climate. That's going to be chapter 2. But nonetheless, I'm just telling you why we're not talking about the European taxa that descended southward. It's because we lost so much of it in the during the Pleistocene, the whole Pleistocene really. And it's because there's mountains going this way in Europe and there's seas here. And so during the last glaciation, when plants were tracking southward, they had nowhere to go. So there was massive extinction in Europe. Whereas in North America and Asia, there were refuges where plants could go to. We'll talk a lot more about refuges in just a little bit. So the study I'm going to talk about, just having that concept in mind that we have Asian and North American sister relatives, right? The study I'm going to talk about is certainly not just my work. It was actually the brainchild of Robert Rickliff's right here. If you ever took an ecology class, Bob probably wrote the textbook. He's sort of one of the fathers of modern ecology. He's a fascinating person. He is now retired. Yvonne Jimenez still does lots of modeling at Missouri Botanical Garden. He's like a master mathematician and from Columbia, is really into Latin American diversity. And then June Wen's at the Smithsonian. So her specialty is studying disunited taxa between Asia, North America, and Europe. So she helped us compile species that we looked at. James Zakos, he's actually a climatologist, and he provided the climate data set that we used for this research. So this is just a sample of lineages. For those of you who know your genera, you got to know your genera. Just kidding. You'll recognize, for example, Aurelia spinosa, devil's walking stick, Aesculus, that's buckeye, Cerasus is redbud, Campsis radicans, can't think of it right now, Liriodendron, you got tulip poplar, witch hazel, all of these that I'm naming tracked southward and had a sister taxa go southward into Asia. Isn't that cool? So if you go over to Asia, you will find these same genera. And usually they're even more diverse over in Asia than they are here. And why is that? It's bigger and the mountains are more complex. So there's more species to occupy all the little microhabitats in mostly China, southeastern China. So yeah, it's just, I think it's fascinating. I haven't been to China yet, but I would walk through the forest in China. Yes. Yeah, good question. So what time frame? So we are talking the disjunction, as they call it in science, in case I say disjunct taxa, that's referring to these species that separated from Asia and Europe. And this separation event started about 90 million years ago, and it continued up until a few million years ago, like about 4. But the most dramatic cooling period was about 50 million years ago up until like 40 million years ago. There was dramatic cooling, so the plant taxa just migrated faster. Of course, it's a slow process. Takes generations for seeds to disperse and track southward. But eventually, over millions of years, you're going to have lots of species moving. That makes sense? All right. So our hypothesis for this study was that the ancestral way in which species responded to climate change is similar to how species that are the descendants of the ancestral taxa, similar to how the descendants respond today. And just bear with me here. I know this is like a lot to wrap your head around, but basically we figured out a way to measure ancestral cold tolerance of those taxa that occupied the northernmost latitudes up here. So ancestral taxa, meaning the common ancestor between the Asian and North American taxa. We figured out the climate that they could tolerate, and I will tell you how in a second. And then we looked at the descendants that went into North America, and we measured the climates that they tolerate. So how do we do this? Well, so measuring the ancestral trait, or basically cold tolerance of the Asian and American common ancestor. So we did this by basically mining the research community for all the papers that we could find, research papers that show the time of disjunction, the time of separation of Asian and American taxa. And this is based on genetics, so the molecular clock. And so this is where June Wen from the Smithsonian comes in. So when you look at genetic distances between two species or two genera or what have you, you can figure out how genetically dissimilar they are and figure out when they diverge from one another. In this case, they diverged geographically, right, into Asia and North America. And so what we did is we used the time that Asian and American taxa separated, which comes from all this genetic literature. It's actually a field called phylogeography. I don't know if you all know what that is, but you're going to learn a lot about it. So we looked at the time, for example, when, let's say, this oak separated, let's say it was 80 million years ago. And then we looked at Earth's paleotemperature, so the temperature of Earth when that Asian oak and that North American oak separated. And that's how we figured out how much cold those oaks could manage. So it's like an indicator, right? It's like a metaphor. And then we looked at, for example, the spruces. When did the spruces in Asia and North America separate? Well, gosh, it was only 5 million years ago. And so they are very tolerant to cold as compared to the oaks. And we did this across 43 different genera. So basically all the research that was out there. And then we looked at the modern day trait. Everybody with me so far? All right. If you aren't, you can tell me. It's okay. Yes, question. Good question. Thank you. So how do we know what the temperature was 80 million years ago? We use oxygen isotope data in ice cores. Now, this isn't me. I'm not mining in ice cores. In fact, it comes from mining for oil. Part of what happens in that process is oxygen isotope data is measured down the core, and you can figure out Earth's paleotemperature. And that's based on like rate of evaporation of water into the air and stuff like that. Yeah. And that's where Zakos, the climatologist, his data set comes in. So you can see really different data sets, right? And they're coming together to figure out some ancestral trait in a really unique way. Not my idea. This was all Bob Rickliff's. I just did the hard work behind it. So the way that we measured the modern trait of the descendants that went into North America is, again, we're capturing climate data. This time we can get annual mean temperature, and this is, of course, data that we can collect today. It's weather station data, essentially, underneath the distributions of species. So each of these dots is a location for one of the 116 species used in the study. And the reason why we went from 43 genera to 116 species is, of course, there's been a lot of evolution. Since those ancestors were tracking southward, they've evolved into more and more species. And we're talking millions of years. So they've at least doubled in the amount of lineages that we're seeing. So 116 species in North America, we capture climate data underneath these species. And we can then, again, estimate cold tolerance. So we did that by getting the northernmost percentile of that species distribution to estimate the species tolerance to cold. So we're looking at just the American descendants, unfortunately not the Asian descendants. And that's because, unfortunately, in Asia, they don't collect all this valuable plant distribution data like we do. You know, a lot of this comes from the Forest Service. Some of you I know have had the Forest Service come trample on your property and set up a quadrant and take data about species. That's where this data is coming from. So thank you, Forest Service. So yeah, these would be the Asian relatives, for example, and we can't do that yet. Maybe someday. That would be really neat to compare how one's evolving over the other. So our results are statistically significant. The ancestral trait or tolerance to cold is statistically significant and correlated to the present day tolerance to cold. And I'm not going to go into detail about these graphs, but basically, these were different percentiles. So the 5th percentile, the 10th percentile, and the 25th percentile. So that's the different, like, northernmost part of the descendant species ranges in North America. And that was just to get an idea of cold tolerance versus, like, looking at the average temperature. We looked at the northernmost temperature across the species range. And then paleo temperature, of course, comes from Zakos's climatology data. And I know that looks kind of like a mess, but you're also talking about lots and lots of error because we're talking millions of years that we're estimating with these genetic studies. And these are giant data sets that we have put together. I mean, think about all of the genetic data sets and then the oxygen isotope data sets. And then we have the current annual temperature data and species distribution data. And it's just amazing to me that we've stuck all this data together and we have this statistically significant correlation, right? And so what does this actually mean? Okay, so that was a fun exercise. It actually means that there's some pretty big implications for how species respond to temperature and how it kind of like stacks up our communities across Earth over time. Because millions of years ago, it was first those oaks coming down. Now we know. And then it was this species being introduced to the community. And then it was this species. So it kind of, it gives us a lot of insight about these big picture patterns where we might see species across Earth because they did respond to climate and are still responding to climate in the present day as they did in the past. It also means that, well, when you think about climate change now, there's only so many ways a species responds. It can, of course, go extinct with climate change. It can adapt, or it can migrate and track habitat over time, and then maybe still adapt, but still track the same temperatures over time so it doesn't have to adapt. And that's what this study is showing support for, that there has been some climate or habitat tracking over time for these species. And again, we're talking millions of years of time, that we're seeing some signature of species responding similarly to the climate. So any questions so far? No? All right. Yes. It is just about cold tolerance. We wish we could figure out more ways to measure an ancestral trait, but usually ancestral traits are measured with fossil evidence. So fossils, with like, yeah, that's the main way that traits are. And so this is just our unique way of trying to get some other measure of a historical trait. Yes. Yeah, so cold tolerance is like, okay, yeah, so where does cold tolerance come into this? So, good question. So, it's like, it's really, we are trying to figure out a word or a name for the trait that we are capturing by using this method. So, by doing this, by figuring out the temperature of the earth, and paleotemperature is measured in the northernmost part of Earth at the time of separation, it's going to be cold tolerance because the reason these things are separating is due to the cold, if that makes sense. So something that has a disjunction time from Asian relatives 10 million years ago is going to be less cold tolerant, trying to get away from the northernmost latitudes, than something with a disjunction time only 1 million years ago. It would have lingered up in those latitudes longer and separated from those Asian relatives more recently. So that's what we looked at, is like trying to capture a modern day trait that is indicative of what we were seeing here. So yeah. So yeah, we were basically looking at paleotemperature, the temperature when these lineages are separating, and kind of assuming they could be separating due to the cold. And if it correlated to the temperatures that the descendants currently occupy, then there's some relationship there between what they could handle millions of years ago and what their descendants are managing today as far as climate goes. Okay, I'm getting some nods and some lots of questions. Yes. Mm-hmm. Yes. That's a great question. That is a great question. So how does it give us any insight about today, when climate change is likely happening a lot faster, when we're talking about tens of years and not millions of years? So good question. And I get into that a little bit more in a little bit, but I will say that what we see, for example, at this thermal maximum right here, gives us some insight in what we might expect and how we might expect ecosystems to be distributed, because Earth was very, very hot at that time. I can go back to this. At this time right here, the Paleocene, Eocene thermal maximum, I mean, it would be a worst case scenario, but if we were to get tropical here and north of here, that gives you an idea. Now, as far as habitat tracking, I mean, it's a good question. We wouldn't expect species with really long generation times to be able to track habitat very fast. because they're so long-lived, they're not dispersing their seed or their fruits fast enough to be able to track habitats. And there could be more potential for adaptation that this study doesn't give any insight about. Yeah. Yes. And I go into that a little bit more later on. Okay. Oh, sure. Absolutely. Okay. Sounds good. All right. So those were some ancient events. And now we're going to talk about more recent climate change. So going into the Pleistocene. Yes. Mm-hmm. Absolutely. Yeah. It's it all happens. So sorry, the question is, he understands that the plants are tracking southward as it gets colder, but it also has to do with the animals. Absolutely. It's assumed that animals are dispersing seeds and fruit, unless it's wind pollinated or unless it's not a animal dispersed fruit. But yeah, that's part of it for sure. All right. Good question. So the Pleistocene in the Appalachians. Oh, it's such a romantic time, right? Who doesn't like to go up on the grassy balds and imagine the mastodons and the woolly mammoths up there, the saber-toothed tigers and the short-faced bear and the giant ground sloth? And we had so much megafauna here, and a lot of people don't realize that we did, but there's fossil evidence for it. And there's evidence just in the ecosystems we have at the tops of the mountains, the grassy balds, right? It's pretty agreed upon that these would have provided grazing habitats for the mega herbivores of the Pleistocene. And that's just exciting to me, right? So of course the grassy balds then would have been co-opted by the Native Americans who were here, who probably kept them burned, maintained for hunting purposes or rituals or lookouts, who knows. And then early settlers maintained the grassy balds further. When they got to North Carolina, they're like, oh my gosh, pasture at the tops of the mountains where we don't want to live. This is perfect. So yeah, grassy balds. Of course, there's lots of giant fruits still around today that aren't dispersed by any animals that we have today. And so those are just giving us more evidence that we had megafauna here, like our pawpaw right here, would have been dispersed likely by a hungry mastodon or what have you. And just while we're on it, there's lots of giant fruits like your Osage orange and your avocado, your apples and your wild pumpkins that would have been distributed by megafauna that we don't have today. And we're so lucky that we had these megafauna because it gave us some lovely now domesticated fruits that we otherwise might not have. So the process of having megafauna probably aided in the domestication of these really large fruits, right? So going back to the climate data, so we talked about ancient climate events and species moving around the Earth. What about more recently? So now we're talking thousands of years before present. So the Pleistocene started about 2.5 million years ago and it went up until 11,500 years ago. And it's characterized by lots and lots of glacial events. Of course, we call it just the Ice Age, but actually there's lots of glaciations characterizing the Pleistocene. And the last glacial maximum, 21,500 years ago. This is just a blip in time, right? This isn't that long ago, really. Would have looked like this in North America. So this is the ice sheet that's coming down all the way covering the northern part of Pennsylvania. and the northern parts of the United States. There would have been glaciers covering much of the Rocky Mountains and Sierra Nevada and Cascades. And again, the northern or highest elevations of the Appalachians would have been very cold, like tundra habitat, and grazed by the megafauna. So tundra grasslands at higher elevations, where were the spruce fir forests? They were at the low elevations. Isn't that mind-boggling? So we had a pretty continuous conifer forest. Now we see it at the mountain tops, you know, our spruce fir forest, which is now the second most endangered ecosystem in the United States after the tallgrass prairie. 21,000 years ago, it was vast. It was across the Southeast. And it actually got squeezed down, the vast boreal forests, you know, all the way from like Sweden and Canada, got squeezed down into the Southeast right here. And there would have been lots of conifer woodlands at our lower elevations where we currently see our oak hickory forests. Isn't that mind-boggling? Some of you are like, I know that. So there was, yeah, lots of squeezing of all these species into these refuges or refugia, if you will. And where did our hardwood forests go? Our lovely oak hickory forests and, you know, the walnuts and the tulip poplar. Most of them ended up at the southernmost part of Mississippi and Alabama in the low Mississippi River Valley in the southeast. There were some that would have taken refuge like along the coastline. Some did even take refuge just underneath that glacier. But the vast majority went to the warmest and lowest area that it could, just above the Gulf of Mexico. And just like Europe, you know, we likely experienced massive extinction because of the Gulf of Mexico at that time. So there's actually evidence of another spruce that was around at that time. We have fossils of a spruce lineage, and that's kind of interesting to me. We think a lot about modern day extinction, but there was lots of extinction in the past too, just saying. And it's kind of interesting to think that species didn't track together in communities. What we know as the spruce fir forest, it didn't track southward like that and stay together. Species migrate independently of one another in response to climate change. So there would have been novel assemblages like your sweet gum actually co-living among fir trees. So pretty wild, huh? Now, the next study I'm going to go into is my dissertation research that was conducted in the western part of North America. So the Rockies, Cascades, and Sierra Nevada. And I essentially did a phylogeography study. And that's just a fancy word for studying the evolutionary relationships within a species. So the genetic structure of a species. And what we can figure out by looking at basically this giant family tree within a species is what barriers there were to gene flow historically. And because the last glacial maximum created such chaos in compared to where species are today, usually the signatures that we're seeing in the genetic structure of a species are a result of barriers that occurred during the last glacial maximum. So literally glaciers that were across the western part of North America separated up populations, and there was no longer gene flow occurring across that glacier. Or there were like novel arid environments that were created. And so there was no gene flow across them. And we see that in the genetic structure when we collect tissue samples and look at a species today. So I decided to pick a species called white bark pine. I didn't realize it had one of the biggest genomes in the world. I guess my advisor didn't know that at the time either, but nonetheless, white bark pine, I also picked it. So I picked it because it is an endangered species. Not only does it occur at the highest elevations in western part of North America, but it's being impacted by a fungus called blister rust, and the pine bark beetle, which is actually native and attacking all of our pines out west. It's like this horrible synergy of climate and fungus and pine bark beetle. This species is not doing very well. It was finally listed just last year, actually, as an endangered species, a federally endangered species. This is the distribution of it. As you can see, it really likes the mountaintops, just like us. And then this is the Clark's nutcracker. So it has a tight mutualism with this bird in the jay family. So the Clark's nutcracker, kind of like our crossbills, has a unique adaptation to pop the pine nuts out of white bark pine. And it's the primary disperser, along with squirrels and bears. The Clark's nutcracker thinks that white bark pine is like candy, kind of like bears thought that chestnuts were candy. I mean, the oaks are okay, the acorns, but so we want to keep the white bark pine around for these poor birds. Like they want their candy. So this is where white bark pine occurs, at tree line. So it's a tree line indicator, a tree line species. And here's just an image of it not doing so well due to blister rust and the pine bark beetle. This is a picture in Wyoming. And so, as I was saying, when we will look at a genetic structure of a species, we can get ideas of historical things that happened in the past, like where forests used to be at the last glacial maximum. And we have a really, before I went into this study, we had a good idea that there may have been a continuous conifer woodland in what is now the Great Basin and the Columbia Basin. So these modern day dry environments where you don't find a lot of trees, thanks to pack rat middens or pack rat nests, we see lots of plant material that indicates there were conifer woodlands in this valley right here between the Rocky Mountains, the Sierra Nevada, and the Cascades. Of course, there's other research that supports that, like pollen data. Just like we take ice cores and get oxygen isotopes, we can take pollen cores and look at the history of pollen stacked in the soil to figure out what species we may see. And again, this is not just me doing this research. This took a whole team of people, particularly the Forest Service. These are all people who either are working for the Forest Service or were about to work for the Forest Service. Shortly after the picture was taken, they got jobs. I almost worked for the Forest Service, but I had to move to Oregon and I wanted to be in North Carolina. So, so anyway, the Forest Service helped me tremendously, particularly with gathering the tissue material that I needed to extract DNA from across the range of this species. In order to get the genetic structure of the species, you have to sample the species from across its range. So I'm not going to go into too much detail here, but basically the 32 gigabase genome had to be mined for differences between the individuals, right? And 20 years ago, people used to have a handful of these genetic markers that they would use across the genome to figure out who's related to what. Nowadays, all genetic studies like this are actually genomic studies because sequencing is so cheap, sequencing a genome. Basically, the process I did is called GBS, or genotype by sequencing. You just chop up the whole genome in a bunch of different ways, and then you match the genome to itself and find differences. So single nucleotide polymorphism. So this is just an area where this sample of tree has a this sample a tree as a G and this sample a tree as a T. So that is going to be one area across the genome where these individuals will differ. Yes. Single nucleotide polymorphism. Bio 101 or genetics 101. All right. So yeah, SNP. So that just means that at this spot in the genome, there was a mutation, right? That's how we get all of our novel genetic material is by usually DNA polymerase screws up and we have a mutation. So so this is literally a screenshot from my computer in graduate school. I have PTSD looking at this right now. I'm just kidding. So these are the tree samples, and I had more than this. There were hundreds of them. Because of course you have to duplicate everything in science. So each population had at least 10 or 12 samples. And then up here are locations across the genome. And so here I'm trying to figure out who's related to what. So again, just matching the G's to the G's and the A's to the A's. And this is how we make evolutionary trees, either within species or between species. And this ties back to the first chapter that I talked about, where I talked about June Wen gathering all the data, the genetics data, and figuring out at what time lineages separated. It's just using this. So using the molecular clock. Yeah. So as I said, the Forest Service helped me tremendously. And they actually kind of told me to choose this species for my research because they're like, we need this information. And I wanted to have a useful dissertation, right? I didn't want to just be sharing it with the research community. I wanted to do real conservation. And so they wanted to know the genetic structure of the species so that they could do the best they could conserving the species. Basically conserving as much genetic diversity of the species as they could. And so fortunately for me, they do inoculation experiments. So for example, this could be a population from Glacier National Park. This is a population from Yellowstone. And they're inoculating these populations with the fungus to figure out what's resistant to the fungus. And so then they breed a bunch of resistant white bark pine, and then they can go plant out the forest with resistant ones. The same thing's happening with our American chestnut trees after breeding some of the Chinese chestnut into them. There's programs where of course, with hemlocks or Eastern Canadian hemlock, where this is happening as well with the hemlock woolly adelgid. And so this made it really easy for me to get tissues across the range of the species because I just had to go visit Durana Genetic Resource Center in Southern Oregon and another place they're doing this in where was I? Washington, Idaho. And I collected a lot of the tissue from the species. Not that I didn't go out in the field a lot and do it a lot that way too, but it helped having this here. So this is just an example of an inoculation experiment where one family, these are very close relatives, are showing resistance. So you can see that green line across the box. Those are all relatives that got inoculated and actually survived. So yay. And what do you know? I'm going to just show you just snippets of the results from my research because there's lots of graphs and I really don't want to bore you, but I'll show you the most exciting stuff. So yes, there was lots of evidence for a continuous woodland because Crater Lake National Park down here, Glacier National Park way over there, Lawson Volcanic National Park, Rainier, the Tetons, and Umatilla, they are on each sides of this Columbia Basin, and yet genetically they're the most similar. So you have evidence here of this conifer woodland that would have occupied and had lots of gene flow right in the center there, right? And then this is like a principal component analysis where you're just measuring variation in the genetic data set, and we see a similar pattern right there. So another pattern which we always expect, pretty much always expect, is this pattern of genetic isolation by distance. That just means in general, those plants that are, or those populations that are more closely, close physically, are going to be more related genetically. And that just comes from modern day gene flow. You know, pines, of course, are pollinated by the wind. And so there's lots and lots of gene flow happening out there, right? They're spreading their seed long distances as the wind blows, versus pawpaws, well, I'm guessing they're pollinated by insects. But anyway, you understand, wind pollinated things have lots more gene flow than insect pollinated. So I will brush over this, but just another way that we figure out where habitats used to be, like in the last glacial maximum, is from climate modeling. And I did this as a third part of my dissertation research. And what we basically do is, again, get all this climate data underlying species occurrence data. So in red is where white bark pine occurs. Again, Forest Service data. That's how I'm getting these occurrences. In blue is where it doesn't occur, because you need these absences to help make these models really robust. And then you capture climate variables underlying this species occurrence data. And you use really complicated, robust models. I got help with this. This required a lot of coding, and it was tricky. And my little computer sat and ran for about a week. I couldn't touch it. It just ran models and ran more and more models. If I would have gone to DC and used a supercomputer or something, it wouldn't have taken very long. But yeah, basically, I ran climate models to figure out, given a future climate scenario, where is white bark pine going to be? And so this is sort of the worst case greenhouse gas emissions scenario. And this is year 2070, which is as far as I projected into the future. And what I found was kind of interesting. So these outlines here are wilderness areas, and they occur usually within broader national forests, right? So this is the Upper Cascades, Sierra Nevada, and Yellowstone. And I found that as the temperature gets hotter, and this hopefully partly answers your question, where are plants going to go? Well, we're doing lots and lots of these kinds of models with different species to try to figure that out. But what I found was that white bark pine used to, or currently, occurs well outside of the wilderness areas. With climate change, it ends up in just the middle of wilderness areas. And you might think, well, that's pretty good. Wilderness areas are highly protected. But actually, with a species like white bark pine, it requires active conservation work, like planting out seedlings that are resistant to blister rust in order to keep this species around. It also requires lots of driving massive amounts of seedlings into the woods to plant them out and everything. And so this had implications about policy, right? We're going to have to update our policy to keep this species around if it's going to end up just in our wilderness areas, basically. Because right now you can't go and plant trees in wilderness areas. That's not okay by wilderness area standards, right? They're meant to be the most pristine. So in closing, everybody, I hope that the next time you are out hiking, you think about big climate patterns and the way they may have shaped species as they're distributed across the earth. It's something that I think a lot about, but and I hope you think about the Pleistocene and the grassy balds, and of course the megafauna and the fruits they may have eaten, as well as the refuges of the plants and where they may have been at the last glacial maximum when there were major ice caps covering most of this continent. And of course, the spruce fir forests having shifted all the way from the vast boreal areas just down into the southeast and creating not only this squeezed little area, but then when the glaciers receded, of course, we got a whole nother fir and spruce species. So our red spruce and our Fraser fir down here, they don't actually reproduce with those in the boreal forests, right? So we have novel species because of all of this. And I'm preaching to the choir here, but of course, I'm a big proponent of having more national forests and more state parks and more national parks. And even the Blue Ridge Parkway is doing its part by helping conservation, because when you have corridors connecting these disparate areas, that's how plants and animals migrate through time. If you have any takeaway from this talk, I hope you understand the vast change that has happened in Earth's history and how plants are going to move over time, and they require areas to move to. And when you have lots and lots of developed and fragmented land, it reduces the potential for them to move, if that makes sense. Yeah. So this is a very boring slide, and it just thanks my advisor and a bunch of people who I used to work with a long time ago. So genotyping, the genetics analysis, the modeling I talked about, collecting plant tissues, Missouri Botanical Garden, the Forest Service, and Washington University. But really, what keeps me going on a day-to-day basis are my nerdy friends. Randy, if I knew you were going to be here, I would have had a picture of you. I'm sorry. I have a good friend back there. But yeah, so thank you to my friends, to my colleagues. Of course, I have lots of pictures of the guides of Asheville Hiking Tours here. Thanks, Eli. She's my manager, and she does an amazing job. And yeah, thank you all for being here. Of course, thank you, Diane Mooney, for inviting me today. This was so special. And to the Blue Ridge Naturalist Network. All right. Oh, okay. Oh, sure, yeah. A few more questions.
We're advised to plant native plants only to avoid invasive species. When a plant has an ancestor that's now in the other half, another hemisphere, would you consider that to be a native plant too? Or has evolution changed it so much that it could be considered non-native?
Ooh, that's a good question. I wish Luke Cannon were here to answer that. I think he's listening. So yeah, I certainly consider plants in Japan and China non-native, even though their relatives are here, right? We have horribly invasive plants because over millions of years of evolution, so much has happened. There's been lots of coevolution where diseases have evolved to keep those plants knocked back. And it's the reason why, I showed a picture of China and how similar it looks to here. It's the reason why Chinese and Japanese plants come over here and they're wildly invasive, because all of that coevolution that has occurred in their homeland hasn't occurred here to keep those plants in check. So yeah, certainly still not a proponent of planting Chinese plants personally, of course. Yes. Oh, okay. Microphone.
From the front to the back and back to the front. I work a lot on climate change presently, and one of the things that struck me about one of your slides is your primary temperate hardwood refuge.
And that's not my research. That is Delcourt and Avis. So that, okay, so sorry.
So the question I have for you, has anybody in your group talked about the fact that that's one of the very few areas in the entire world that did not warm very fast? That area, it's very unusual. It's been trying to figure out why that's the case, but that's one of the few areas in the world that did not warm. It's starting to catch up, but it's still well behind. And it's very anomalous. And I just find it fascinating that that was where your primary temperate hardwoods decided to refuge.
That is very fascinating. So I immediately go to thinking about the oceans. I know that oceans tend to keep temperatures mild and regulate any extremes. And of course, this Mississippi Delta area is going to be, there's a lot of water and it's really complex. But that's fascinating, like just in a bigger picture and yeah, bigger patterns being painted. Thank you for letting me know. Yeah, anybody need a topic?
It may be an embarrassingly basic question, but on your Pangea map, there was terminology about Bombanian and Wasatchian and Apraesian. What does that refer to? Or is that a different name for the landmass or what grew on it?
Oh no. Okay, I probably picked a slide just because the picture was the best and had the highest resolution because I have no idea what you're talking about. Okay. Um. Oh. Oh, Laurasia and Gondwana? Oh. Oh. Yes, flightless birds. So I did it. This was the only image I could find that was a good image of the land bridges. And so yes, this is from another research paper about flightless birds. That covers the fauna of today's talk.
Hi, at the risk of getting a bit too specific, I'm a big fan of the pawpaw.
Oh yeah.
And the story, as far as I have grasped it, is that somewhere back, I don't know when, the Asimina triloba, the American pawpaw, who has there's nothing else in that whole genus or family in the United States, but much further south, it's actually in the custard apple family. And I think that's Caribbean or maybe even elsewhere, but very much further south. When did Asimina triloba separate? What happened to it over time? It got a cold gene from somewhere. It got a cold adaptation, but when? And how far did it get south? And are there any pollen remains or anything up further north even than its current extent?
That is a wonderful question. And I don't even know that somebody's done that research. I can tell you that we did not include that in this particular study. So I'm guessing that the evolutionary history may not be well researched, but I'm not sure. I don't know. I'm sorry.
So the family, I'm not even sure. Asimina is the genus. I'm not even sure where the rest of that genus is, or if it's even a single species genus. But the family is custard apple, that I know.
Yeah.
Oh well, it's a fun fruit.
It is a fun fruit.
I've got 4 trees in my yard, and they produce well over 100 fruits each year. So
I actually horribly scheduled my talk today, and I closed on a property this morning where I had planted pawpaw only 2 years ago, and I'm not going to be able to get to eat it. I'm so upset, but maybe I'll go raid it someday in the future. So, it takes several years for it to fruit.
6 or 7, yep.
Yeah, double book my schedule just a little bit.
One more.
All right. Any more? Oh, on Zoom. All All right. Are we good? Well, yeah, thank you, everybody. Thank you. Yay. Oh, thanks. Appreciate it. Yeah.